2.
This is John Kim, AP Bio Video, Period 5.
Harrison Lee, Period 5.
William Park, Period 4.
Etienne Harpainer, Period 4. And we're doing AP Bio Lab 4, photosynthesis.
The purpose of the lab was to measure how different conditions affect the rate of photosynthesis through a calorimeter. Through boiled versus unboiled, light versus dark, the photosynthesis rate will be measured because light drives photosynthesis. The light excites the electrons, which are accepted by the DPIP. The DPIP goes from blue to clear, meaning that the percent light transmittance is increasing. The control was just the max mix, which is the phosphate buffer with distilled water. The first cuvette, which is unboiled chloroplast, and master mix will be used to calibrate the sensor. The hypothesis is that the more light shine on the unboiled chloroplast suspension, the greater percent light transmittance.
Here I'll be giving you more background information. The focus of this lab was photosynthesis, the process by which autotrophs use energy, carbon dioxide, and water to create glucose and oxygen. Photosynthesis occurs in certain organelles called chloroplasts. Chloroplasts contain pigments called chlorophyll, which absorb various wavelengths of light. Chlorophylls differ in the wavelengths that they absorb. Photosynthesis requires light energy to occur because when light hits the chloroplast, electrons become excited and passed along an electron transport chain. The electrons are then passed along the electron transport chain in the thylakoid compartment of the chloroplast until the excited electrons reduce NADP+ to form NADPH. Photosynthesis uses the electrons in their excited states to create ATP before the electrons drop down back to their ground state, releasing light and heat in the process. Photosynthesis also uses two types of photosystems, which are chlorophyll and proteins. Photosystem 1 absorbs light with wavelengths of 700 nanometers to excite its electrons, which are used to reduce NAP+ to form NADPH. Photosystem 2 absorbs light with wavelengths of 680 nanometers to excite its electrons, which move along an electron transport chain to fill the missing electrons in photosystem 1. Water splits as its electrons fill the missing electrons in photosystem 2. These two photosystems are important to photosynthesis because they work cooperatively to form ATP and NADPH. However, in this lab, the excited electrons are taken by the DPIP.
Each of the 5 cuvettes was filled with different contents. One cuvette was filled with just 5 milliliters of master mix and unboiled chloroplast and was used to calibrate the calorimeter, which means that this cuvette was used to tell the calorimeter what 100% light transmittance was. The second cuvette was filled with 5 milliliters of master mix and 3 drops of unboiled chloroplast suspension and covered in aluminum foil. The initial percent light transmittance was found to the calorimeter. The third cuvette was filled with just 5 milliliters of master mix and 3 drops of unboiled chloroplast suspension, and then the initial percent light transmittance was found. The fourth cuvette was filled with 5 milliliters of master mix and 3 drops of boiled chloroplast suspension, and then the initial percent light transmittance was found. The fifth cuvette was filled with only 5 milliliters of master mix, and the initial percent light transmittance was found again. This cuvette was the control. After the initial percent light transmittance was found for each cuvette, the second to fourth cuvettes were taken to the stand where light shone on the cuvettes for 5 minutes. After 5 minutes, the percent light transmittance of each cuvette was measured. Then the cuvettes were taken back to the stand for another 5 minutes. After another 5 minutes, the percent light transmittances of each cuvette were found again. The cuvettes were taken back to the stand again for another 5 minutes. After 5 minutes, the percent light transmittances of each cuvette were found again.
Now that William has described the methods, Harrison will now demonstrate the lab.
Okay, so here we'll be telling you more about the methods. Alright, first of all, the dependent variable was the percent light transmittance, and the independent variable was time in seconds. And here's the control. The control is just 5 milliliters of master mix, no DPIP, no chloroplast suspension. Here's cuvette 1, which is meant to calibrate the sensor to tell what 100% light transmittance is. and this is just unboiled chloroplast suspension and master mix. There's no DPIP in here. So to calibrate the sensor, we wash it with a chem light. We put it into the sensor and press this green button, and the light will be green, and when it comes off, that's when we know that the sensor has been calibrated. Alright, now it's been calibrated. Okay. So this is cuvette 2, the unboiled chloroplast in dark run. Inside the cuvette there's DPIP, unboiled chloroplast suspension, and master mix. Okay, so to measure this, I will take out the aluminum cover, wipe it with chem light, put in the sensor, turn on data CO for 5 minutes, and okay, so we're back, and this is cuvette 3, the unboiled chloroplast suspension and light, and there's DPIP, unboiled chloroplast, and master mix. So we put it into the sensor, close it, or wait, hold on. We wipe it, then we close it, put it into the sensor, stop the run for 5 minutes, and we'll be back when 5 minutes are up. Okay, so this is cuvette 4, the boiled chloroplast suspension in light. Inside there's boiled chloroplast, DPIP, and master mix. Okay, so we'll wipe it with the chem wipe again. We'll put it into sensor, and we'll be back after we start data C for 5.
Hi there, this is John Kim with the results. The percent light transmittance of the dark cuvette stayed the same for the most part. The percent light transmittance of the light cuvette quickly increased in percent light transmittance when compared to the boiled cuvette, which slowly increased in percent light transmittance. The control cuvette, phosphate buffer distilled water, stayed the same for the most part. The hypothesis was correct because more light shown on the contents in the cuvettes led to a greater percent light transmittance. The boiled chloroplast suspension's percent light transmittance slowly increased when light was shown on them because the chloroplasts were denatured, so the function changed. The boiled chloroplast suspension's percent light transmittance also increased because of the specks of chloroplast settled down as time progressed. The unboiled light chloroplast covered in aluminum foil would have had a greater percent light transmittance as time progressed, but light could not get in, so electrons could not be excited. Thus, the DPIP could not receive the excited electrons, so the color of the suspension remained blue throughout the experiment. The unboiled chloroplast suspension percent light transmittance increased rapidly because light energy was able to excite the electrons of the chlorophyll systems in the chloroplast. Thus, the DPIP was able to take away the excited electrons, and the color changed from blue to clear. The DPIP cuvette's light transmittance stayed the same because there was no chloroplast added to this cuvette. This was the control, and as expected, virtually nothing happened to this cuvette's light transmittance. In this experiment, the increased light transmittance signified that the rate of photosynthesis increased because the DPIP went from blue to clearless when gaining the excited electrons. The excited electrons came from the electrons of the photosystem 1 when they were excited by the light, and the photosynthesis was able to occur. Therefore, the percent light transmittance was directly related to the rate of photosynthesis. Another way to conduct this experiment and achieve the same objective would be to place oxygen sensors around the plant inside a closed container. The students could measure the oxygen levels inside the containers under these conditions, one containing unboiled chloroplast suspension in open light, the control, the same in a dark closet, and one containing boiled chloroplast suspension in light. Measuring the O2 levels would be an indirect way of measuring photosynthesis because a byproduct of photosynthesis is oxygen. Errors could have occurred if aluminum foil did not properly cover the cuvette, which could have allowed light to enter. Another error that could have occurred is if the colloid of the spinach blocked or refracted light.
If you want to know how a plant grows, it takes water, air and sunlight and makes cellulose. Every plant can do this fundamental process, and we can call this photosynthesis. Unlike me and you, plants need CO2, and they make oxygen that stops us turning blue. Every plant can do this fundamental process, and we can call this photosynthesis. It's a miracle how all the chlorophyll capture